All right, welcome everyone to our 8th annual Wisconsin Alzheimer's Disease Research Center's Alzheimer's Disease and Related Dementias Research Day. And it is so nice to be back in person. My name is Barb Benlin, and I'm a professor in the Department of Medicine, and I lead the research education component of the Wisconsin ADRD, which hosts this meeting. So it is really nice to see you all today. We've faced several challenges over the past 2 years, professional challenges and sometimes personal challenges as well. And what really strikes me about our strong group is that nobody stopped working on Alzheimer's disease and related dementias. And we persevered in the research. And all of the work that you all do is so important. And I am just so happy that we're all here together today and that we can celebrate these achievements and all of the scientific progress that we've made, which serves families with Alzheimer's disease and related dementias. So thank you for everything that you do. This research day is designed to encourage collaboration and promote scientific thought among faculty, students, and researchers from a wide range of disciplines across the UW campus. We have an excellent agenda today. So it is up here, and you can see that we have an amazing lineup of speakers. We have a wonderful keynote presentation, and we're really looking forward to the rest of the day. I would like to now take the opportunity to introduce Dr. Sanjay Asthana. Dr. Asthana is the chief of the Division of Geriatrics and Gerontology within the Department of Medicine and holds the Duncan G. and Lottie H. Ballantyne Chair in Geriatrics. He is also the Associate Dean for Gerontology at the UW School of Medicine and Public Health and Director of the Geriatric Research Education and Clinical Center at the William S. Middleton Memorial Veterans Hospital, and the founding director of the Wisconsin Alzheimer's Disease Research Center. And as you know, Dr. Asthana has built a really remarkable program in Alzheimer's disease and related dementias at the UW-Madison. So we are welcoming him to the stage now to deliver the highlights for year 13 of the Alzheimer's Disease Research Center. Thank you, Dr. Asthana.
All right. Good afternoon, everyone. How are you doing? Wonderful, wonderful. Well, thank you. Thank you for being here. I want to welcome you to this research day, and I also want to welcome those who are joining in virtually. I believe there's a good number of people who are listening in virtually as well. So as Dr. Benlin shared with you, the ADRD here has been in existence since 2009. So we are in 12th year of funding at University of Wisconsin, and it certainly has programs that go across the full breadth of Alzheimer's research. It's not just Alzheimer's, but we also focus on other neurodegenerative diseases. So what I plan to do today is just to give you a very brief summary of what's happened in the past 1 year, and also just give you a few highlights of the center. So I need to coordinate, and usually at my age, coordination is a problem, but hopefully it'll work well. So we are one of the 37 ADRCs that are funded by NIH across the country. And you see the US map where these centers are. The dark blue are the ones that are full-fledged P30 Alzheimer's centers across the country, and you see the light blue color there. There are 4 of those. Those are kind of preliminary centers funded by the NIH with the hope that they will over time mature into a full-blown ADRC. So they had some strengths and programs, and NIH wanted to give them some funds so that they can grow into a full ADRC. So we are very fortunate to have 37 NIH-funded centers of excellence in the United States that focus on Alzheimer's and other dementing disorders. You'll notice that in the mountain area, there's none. So the hope is that one of the universities there will soon put together some efforts and get a center in that part of the country as well. These centers not only do research, cutting edge breakthrough research, but they also provide patient care. Indirectly, at least, these centers sponsor memory clinics. So it's important that these centers be available throughout the country uniformly so that patients in each part of the country can receive the best care those with dementias. So the ADRC has, our ADRC has 41 investigators, and they are spread all over the university campus, and they bring in expertise across, once again, all aspects of Alzheimer's disease and other demanding disorders. And we support a number of studies at any one time, at least in the last year, or currently, in fact, we have about 66 active studies ongoing. Many of these involve patients and participants, some healthy controls, and others are lab-based, animal-based studies. So we have programs that merge lab-based research with patient-based research. And that's critical, right? We also have a large pool of participants who come from across the state of Wisconsin, surrounding states as well. And there are currently about 1,100 such people who are doing research in our ADRC. Some of these have Alzheimer's disease, others have a condition called mild cognitive impairment, and many are healthy controls who are at risk for the disease. So we have a variety of people who come and do research at the ADRC. And also these people, they provide a lot of data, cognitive data, physical exam data, medical data. They participate in brain imaging studies. They also undergo lumbar puncture. Believe it or not, about 45% of our participants have undergone lumbar puncture. We have CSF samples on them, and we do all kinds of analytical studies on the spinal fluid. So we have a collection of extensive database and biospecimens, and these biospecimens are linked to the clinical data. So anyone of you interested in analyzing our data, which is huge, then you just submit a request and we can provide you those data and some of these specimens as well. And the ADRC also does a lot of educational events throughout the state of Wisconsin for lay public, for scientists, and for students and trainees. So the education takes place across the spectrum of the disease. In the last year alone, we were very fortunate that NIH gave us about $37 million of grants that focus on 9 different studies. And overall, our active funding is close to about $160 million. So we are very fortunate that NIH believes in research going on at UW-Madison, and they support our research. We have about 85 trainees, all levels of training, who are working within the ADRC system and different programs, and they're getting the training that they wish to get, and hope is that they'll all become future leaders in Alzheimer's research. Many of them are PhD investigators, some are physician scientists, and learners at undergraduate, graduate, and doctoral levels as well. And then we have a clinical core I mentioned earlier, about 1,100, nearly 1,100 people now in those studies. Also, about 66% people have provided consent to donate their brain when that time comes. So we actually have a large brain bank where we perform autopsies and we examine the brain tissue of people who have Alzheimer's disease and those who don't. And about 80% of the participants have at least provided one MRI scan, multiple follow-up scans, and lumbar puncture as well, along with many other data. The quality of people who work here at UW-Madison in the ADRC is recognized internationally. So we have had some very important appointments in the last year. Dr. Cynthia Carlson, who is the director of the Wisconsin Alzheimer's Institute and also director of the clinical core of the ADRC, was just appointed by the U.S. Secretary for Health as chair of the Advisory Council to the National Alzheimer's Project Act. If some of you have never heard of the word NAPA, or the National Alzheimer's Project Act, that was signed by President Obama in 2011 through public law. And what NAPA, for the first time in the United States, unlike Europe, most of the European countries had a national project to tackle Alzheimer's disease. In the US, it was first time in 2011 that President Obama signed a Project Act for a national strategy to address Alzheimer's disease. And Dr. Carlson is chair of the advisory panel to NAPA. So you can imagine how prestigious this appointment is. And we are absolutely delighted and honored that she was selected from UW-Madison by the US government to chair this council. And also Dr. Carrie Gleason is very well known in her research for health disparity. She is really renowned for focusing research on African Americans, American Indian participants, and she just received a few weeks ago the Vilas Mid-Career Award from University of Wisconsin. This is another prestigious award. And also, please tomorrow watch PBS Nova. There's going to be a film on RAP registry. It'll be shown nationwide and probably international audience as well. And they'll talk about research ongoing here at UW-Madison involving the RAP registry. And also Dr. Amy Kind and Dr. Benlin was involved, and Dr. Andrea Bykowski-Gilmore. They just recently founded the UW Center for Health Disparities Research, and this is focusing on how Alzheimer's disease may differ between races and ethnicities, and how poor neighborhoods and poor socioeconomic status may influence risk for Alzheimer's disease. This is a very large area of research called social exposome, and how the sociocultural and other behavioral factors increased risk for Alzheimer's disease. It's a very exciting area of research, and Dr. Kine and Dr. Benlin and their group is really renowned and leaders in that area of research. Also, a number of applicants got the developmental program grant, and these grants are there to help us launch some pilot studies to get some data so that they can apply for larger grants with NIH. The REC scholars, and Dr. Bhattacharya is sitting here, one of those, also received funds from the ADRC for their project. So the ADRC provides funds for early stage investigators to launch their studies and their careers in Alzheimer's with the hope that they will become leaders in the foreseeable future. Dr. Okonkwo, this is I think my second last slide. I might have exceeded my time, but very soon it'll be over. Dr. Okonkwo is again internationally renowned for his research in exercise intervention resilience to Alzheimer's disease. Some people have all the pathology in their brain, but they don't have the symptoms. How come they resist the clinical presentation of the disease? He's doing research in that area. Exercise is one of the means to increase resilience, and he's also leader of a minority recruitment group in a large national center. And Dr. Konopka just recently got an early stage investigator grant from NIH. He's the lead investigator to look how one of the rapamycin analogs called you know what it is, everolimus. Okay, I tried all of the morning, but couldn't succeed here. Anyway, this is one of the agents that is a analog for rapamycin, and it may affect the risk for Alzheimer's disease, cognition, etc., in a beneficial way. So he's one of the first investigators to do a human study and an animal study as well. And finally, a number of grants are being resubmitted to NIH for renewal. And Dr. Johnson is going to lead one of the largest grants that will be submitted in July with the hope it gets funded. That will be the largest grant ever funded by NIH to UW-Madison. So we are keeping our fingers crossed for that grant. We have podcasts. Some of you may have listened to them. Dr. Nate Chin here has recorded about over 150 episodes, I believe 115 episodes, and these podcasts have been listened all over the world, 110 countries, and every state in the United States. So these are podcasts on all aspects of Alzheimer's disease. If you get a chance, please listen to them. They are very, very informative. So with that, I want to thank you again for being here. Hopefully you'll enjoy the day and learn something new about Alzheimer's. So thank you.
All right. Thank you, Dr. Astana. And also, yes, please do check out the podcast, and you can actually hear from our keynote speaker, Dr. Karikari, who was interviewed by Dr. Chin. So I would like to now introduce our keynote speaker, Dr. Thomas Karikari. He is an assistant professor at the University of Gothenburg, Sweden, and also holds an appointment at the University of Pittsburgh. He received his PhD in 2018 from Warwick University in the United Kingdom, funded by a studentship from the Biotechnology and Biological Sciences Research Council. As well, at the end of his PhD, he was a visiting scholar at Harvard Medical School with a fellowship from the Royal Society of Chemistry and the Biological Sciences Research Council. His research is focused on understanding the processing of neurodegenerative proteins in the human brain and biofluids, and to apply this knowledge to develop new diagnostic tools for clinical use. And his career has really been on the rise. He's discovered and developed tools to measure specific forms of the pathologic tau protein in the blood. And this is a major achievement in the field, but also a real benefit for individuals who are at risk for developing dementia, for the purposes of early detection, enrollment in clinical trials, and clinical trial outcomes. So this is having a major impact on our field. The first blood test he developed is now commercialized and is awaiting FDA approval for clinical diagnostic and clinical trial use. He's received several major awards for these discoveries, including the Astrup Prize in Clinical Chemistry from the Nordic Federation for Clinical Chemistry and the Best Alzheimer's Publication Award from the Swedish Alzheimer's Foundation. He holds research fellowships from the Swedish Research Council, the Alzheimer's Association, and the Bright Focus Foundation. He's also the principal investigator of several other research grants and has published over 80 peer-reviewed articles and also has an awarded patent. So we are so pleased to have him join as our keynote speaker today. Please join me in welcoming Dr. Karikari.
Thank you very much. It's I wish I could be there because with the screen showing the auditorium and how I could see all of you, it's just amazing. And so yeah, first of all, pardon me that I couldn't be there in person to be with you, and I do promise I will make it to visit Wisconsin if I get the chance in the future. And also I told Babs that this is going into the books because this is the very first keynote invitation I have ever received. So it's a pioneering moment for me. So good evening from Sweden, and sending regards from all the big Alzheimer researchers here, from Henrik Zetterberg and Kai Bleno and all the other folks that we work with here. Great. So yeah, we're going to be talking today about blood biomarkers that seem to be taken to be on the horizon more recently, and then I'm not going to bore you with how exciting it is and how the new discoveries leading to dozens of papers. But also what I want us to look at today is more, let's say, quite more of a very critical look that with how far we've come, and then where should we be looking at next, and all the things that are ahead of us. What are the critical questions we should be asking, and also to ensure that when it comes to the clinical use and how we interpret these biomarkers, we have them in the right way. Great. So as we know, Alzheimer's and they're very symptomatic in focus, and that we're looking at biomarkers or looking at a very biological description of the disease, and that's how we can move forward in terms of providing very biologically focused understanding for the disease. And that for biomarkers, we move from pathophysiology, and then there's PET biomarkers, and that we have CSF, and now we're looking at blood. So I'm going to rush through these because this is the audience that understands this very well. And yeah, so before moving to blood, I was trying to talk a little bit about CSF when it comes to like the ATN criteria with the amyloid, tau, and neurodegeneration. That one of the papers that's kind of my go-to papers was this paper in 2015 in Brain that looked at the Swedish population registry and tried to link all the people who had undergone biomarker analysis in our lab here in Sweden, because it's a focal lab for basically half of the country, and pulled all these data together and tried to provide an understanding that when you look at in CSF for amyloid beta, you get to see that basically for those with Alzheimer's disease, this was just even looking at A beta 42, you get to have the levels that do pack up, like those with early onset or late onset or mixed dementia, mixed Alzheimer's and other forms. Then you have like similar levels, but then it tends to perform quite well, although the ratio later came to show that the ratio provides a better estimate. And that for phosphorylation as well, like when it comes to T, the tau marker in the ATN criteria, focusing on CSF, that you get to see that phosphotel reflects a better understanding, like, when it comes the same way with Alzheimer's early, late, and mixed, you have phosphotel really packed up there. And in other forms of dementia, the levels were not as much. And this was like a large data set showing that, and total tau doing quite a good job to an extent also. So in as much as we know of the pathological diagnosis, and then we also have PET biomarkers, and then with CSF, and I'm kicking off with CSF because it's more in the line of what I'm going to talk about today. Then the issue becomes, why do we need blood? Because if we're going to talk about blood, what becomes the essence of it? And I like to pivot to the idea of even in these biomarkers, and I use the expression that the availability of access, in terms of access to biomarkers at present, it's an issue. And we often talk about in other parts of the world, which is true, but you would agree with me that even in places like the United States and also in Europe, it's not everyone who gets to have access to biomarker-focused diagnosis. So, for example, if we're looking at throughout the world and provide an estimate that it's about, roughly about 20% of the population that do have access to biomarker-based diagnosis. And even this is a rough estimate, because if you look at the population of the continents, like the US, the Europe, and in Australia, even these ones, it's not guaranteed that everyone's going to have access. And that also, if you take it the other way around, it means that if you take every 5 people in the world, 4 of them will never have access, because in many parts of the world, this form of biomarker-based diagnosis doesn't exist. And that also would include some part of some people in this part of the world as well. And that if we were to move to blood, that would provide a much, much more improved access to diagnosis. And that also would provide us with better estimates, to be able to, more confidence, to be able to say that this disease that we're looking at is Alzheimer's, or it's not Alzheimer's. There are massive epidemiological studies that talk about dementia. It would be lovely that if we're able to move to the next step of being able to say that not just dementia, but what forms of dementia, or at least be able to say this is Alzheimer's form of dementia and differentiate it from other ones. And that would be a lovely thing to be able to do. And then some of the potential benefits, and I'm sure we are aware of these ones, but just to look at, we could be able to look at the confirmatory diagnosis of what is Alzheimer's. For example, you'd expect, like, a lot of the phosphotel markers have performed quite well, that in those who do not have a amyloid positivity in their brain and also don't show any sign of cognitive impairment, the levels of these phosphotel markers tend to be low, and then they kind of increase going up. And that with differential diagnosis also, you would have Alzheimer, those with Alzheimer, especially the dementia phase, will have higher levels compared to those with other forms of dementia. So you can improve that. And one thing that I'm also quite passionate about is population screening. That, as we know about the disease cause, there's also a lot that we need to do in terms of improving our understanding of it in people without symptoms as well. And that would be able to increase when it comes to clinical trials. There's been a lot of talk about enhancing diversity, and not diversity, not just when it comes to skin color, but in diversity in terms of the people who are enrolled in terms of their demographics. And that's one way to do that, will be with blood biomarkers. And this links to this, that we provided an estimate recently that, based on, and we use one of the largest, most well-used cohorts, ADNI, to say that based on our estimate of evaluating ADNI, what we found was that if just a simple estimate, like if you need 1,000 individuals to go through to be A-beta PET positive for a trial, then, and if we're assuming a 20% positivity, then if you do a pre-screening with plasma phosphotel, that when we estimated that about 40% would be positive for A-beta PET, then if you go just by a PET, then you have to, even in the simplest of terms, look at 5,000 individuals to get 20%. But then if you go in a different approach, where you start with blood and to pre-screen, then you cut it down. And not just a matter of cost, but also the matter of logistics, and also the convenience on the people that you would ask to participate. And it's much easier, I think, to have a lot of minority people, and also people who are less likely or to enroll in a clinical trial or any form of research study to consent to giving out blood than other forms of assessments. So let's roll just a quick update on the ATN scheme when it comes to blood. That with the ATN that we know in with PET and with CSF, that with blood, we'll start with the A with plasma A beta. There's when you look at the CSF A beta with different methods, like if you look at the IPMS, the mass spec methods, and then with some of the immunoassay based methods, for this from Alexis, you get to have like a near perfect correlation. So very good agreement between them. But then it starts getting quite fuzzy when you look at plasma A beta methods, kind of to suggest that there's a less agreement between them. And also a more recent study even showed this better, that they compared 8 different assays, 8 different plasma A beta assays, including those that were IPMS and those that were immunoassay forms, that on the same sample set, you could see that the performances were quite variable, and also the agreement between these methods performed on the same individuals, the same sample sets, were also quite variable as well, to suggest that there might be different pools of A beta that these assays do target, and also to suggest that some will perform better than others as well, which makes it which is something that we have to take into consideration when choosing an A beta assay in blood. I'll jump off to the N in blood because I'm going to focus on the T. When you talk about the N, it's more of plasma NFL, because I'll come back to talk about total tau quickly. NFL kind of increases quite nicely in the disease continuum. It correlates quite well with the disease severity. And also there's good correlation between CSF and in blood, because NFL has been a component of the neurofilament cluster of proteins. But one thing that we do understand also, NFL is increasing a multitude of conditions, which is one thing that makes it like a super biomarker. But it would be nice if we're able to get a total tau assay that would perform really well, like in blood, similar to what we have in CSF, where the CSF one, with the exception of some other rapidly progressive dementias, being quite specific to Alzheimer's. So yeah, plasma total tau has not had a good name so far. That some of the initial assays were quite beaten up. So for example, in one of the early papers from our lab led by Henrik showed that if you, in the initial assays that they got, basically you have the same levels in control in MC and AD. You could get some significant differences, but from a biomarker perspective, you're not getting anything because you're not separating between them. And that when they went ahead to measure in ADNI, when they ran the whole ADNI cohort, saw the same story, that there was no separation between them. You could see statistically significant differences, but then from a biomarker point, you could not separate between them. And what we think, and also there's no good correlation between total tau in plasma and in CSF. And one thing that we get to think here is that it's because the pool of plasma total tau that's being measured and the pool of the CSF ones are different, in that the assays that do target the plasma ones also get you have a lot of peripheral contamination, because tau is quite highly expressed in forms of in peripheral tissues also, that may release into blood, and that could contaminate what is coming from the brain as well. Now, a recent paper showed that quite nicely, showing that, suggesting that about is only 20% of the total tau in blood that comes from the brain. So it means that we have to look at assays that specifically target these forms. I'll try and come back to that. Well, more recently, there's I think it just came out last week or so. In our lab, there's been like a focus trying to improve this, and we developed, led by 2 postdocs, to develop 3 different assays, looking at different fragments of tau. That, if you take this as the whole fragment of tau, just looking at the N-terminal part, and they got one form that they call NTA, that in a very preliminary stage, it showed quite improved performance. So this is the new assay that they had, and when you compare that to the Quanterix one in a small sample set where you couldn't see a difference, you kind of can see a separation here. And also if you compare with the Quanterix one and in another sample set, you kind of could see here some separation compared to that. Although the sensitivity of the assay is not what we would love to have, because it's very packed at the bottom, that we would love to see like a better room of separation. And this shows that there's a room to be able to develop assays for total tau in blood and to resurrect the total tau story and get better N marker in blood. And we have been focusing on looking at brain-derived form of tau, and we have been successful at that. Although I can't talk about much of that today because I'll be going through the process of intellectual protection. But that is something that potentially could be out like in the next few months or so. So when we talk about the T marker in blood, where it's the phosphotel, which seems to be like the most one of the most talked about and the most exciting stories in the field in recent times, I try to look at what are the different assays that we have. There's been a few like 181, 217, 231. We laugh about it in the lab to say that people just come like just tell what number you want and then we just recreate it for you. But it's been quite exciting that things that we didn't know, things that we didn't understand, that suddenly we got them to work and we've been able to get the recipe. And also one good thing is that there's a good correlation, good agreement between what is found in one lab and what is found in another lab, which from the start we did not understand. So the mostly assays talk about here, phosphotel 181, 217, 231. There's a question of which one is the best, which increases first, which is what. Well, I'll try and answer that today, although there's still a lot of caveats there. And then more recently, there's a 235 that was reported from our lab. In CSF, there's a blood assay that's being developed. It's not done yet. And then there are mass spec-based immunoprecipitation mass spectrometry assays where they measure all of these together with other sites, including like 199, which was talked about like a decade or so ago, and then 202, 205, which is more a very pathologically relevant site, and 396, which goes quite far off into the end of the protein, is also more involved in tangle formation as well. So in terms of the performance, and this is one of my go-to studies and slides, because I when we started with like the phosphotel story, and as we like to do a lot to be quite critical of the work that we do, we went back to ask that, really, is this real? Like what we're seeing, is it really real? And this was the very first new pathology cohort that we run. And this was also like a chance cohort that they were being monitored for a different story, but people gave blood and also donated, signed to donate their brain when they passed on. So this was a longitudinal study where you had people like 8 years, 4 years, and 2 years before they passed on. And then I remember I ran this study, and then the way we do it, we blind ourselves to it. If we do it here and also if we do elsewhere, like we collaborate quite a lot with the RAP cohort in Wisconsin, we send the data off, and then so like there's no potential conflict there. But then when you plot based on the clinical diagnosis, and these are the coding. So the red is those with AD pathology at postmortem, those with non-AD pathology at postmortem, and control, so those with non-neurodegenerative causes. If you plot according to the clinical diagnosis given during life, you couldn't find anything interesting there. But then if you split and look at it based on the diagnosis at death, at autopsy, then that is where you get to see that the performances are quite good. And even that, we found that those that kind of overlap, especially those in the non-ADs that overlap with the ADs, when you split them up, you could get to see that those ones had secondary diagnosis of AD. So it means that some of the levels always are going to be high when you have AD, regardless of what else that the individual might also be having. And we tried more to look at more pathology diagnosis and longitudinal data sets. And more recently, we collaborated with Professor Galasco at UCSD in a longitudinal cohort, and it's been quite interesting. We looked at like all the three, so all the biomarkers that we have available in the lab, and it suggests that if you look at the pathologically defined grouping, it's only the phosphatels that kind of showed some good performance, like low to moderate to severe pathology, and that when you have the A beta ratio, total tau, and NFL, they were practically flat. And that we saw the same thing when we split according to CIRAD, and plaque scaling is the same thing that we did find. And also when we did the same thing with Bragg staging, it was the same thing that you get to see the phosphatels pick up quite nicely, but the others, at least in this data set, we're not seeing pretty good performances. And to look at how they predict cognitive decline was quite a nice way that we saw. And to me, I'm choosing these studies because there's a lot to choose from. But then comparing blood taken years before someone passes on to when the diagnosis at autopsy, which is the primary, the surest way to say someone has Alzheimer's, it says a lot. It means that what we're seeing is real, seems to be real, and that it gives a very good insight into the disease, and that we can start to use them. So let's quickly look at it like Phosphotel 231, 217, 181. What does it mean, and which one goes first? So we did this preclinical cohort recently with a group in Barcelona, the Alpha Plus cohort, and then we tried to measure all three forms, and then try to model according to the changes that would happen. So like this is based on like disease scores, and then you model according to CSF A beta ratio changes, and basically it would show that it seems to be 231 that crosses the 2SD line, a threshold earlier, and then quite closely followed by 217, and then you have all the others also following up, and that in what they call the low burden group, because the low burden is those who are CSF positive, CSF A beta positive, but then CSF A beta positive, but then A beta per negative. So like that narrow window before you become full blown A beta positive is also basically showing the same thing here. That it's just like 231 was picking up, and then 217 followed up, and then the rest did follow. And then we more recently have also looked at it in a longitudinal setting, all three in a different cohort. And what it suggests here is that although it appears 231 comes in first, and then 217 follows quite closely, longitudinally 217 takes over, and then it keeps going, because it seems at some point 231 peaks. And I think this agrees quite a lot with some of the old data from pathology studies, showing that 231 is one of the first ones that comes on in the pre-tangle stage. But then right about the time that you go into the mature tangle stage, 231, you don't see it again. The reactivity goes down. And then others also pick up. So it kind of makes sense. It seems like 217 follows closely, but then for longitudinal monitoring, it picks up quite well, and the others don't really see any change as well. However, I must say that there are always caveats to this, because we've also seen a data set that's currently in review, where we looked at all the available phosphotel assays from companies and from research labs. It was like a mega, I would say a mega head-to-head study. Basically, what we see there is that there is, for example, a 181 assay that performs for, and this is a setting with like those with controls and those with ADs, like clear-cut controls, AD, non-AD dementias in a memory clinic setting. And what that showed is that you have 231, 217 performing really well. However, you also have like a 181 assay that performs the same or even better than some of them, which in a way agrees with some of the other data set that people have published, to show that if you're looking at it for diagnosis, and I think I have that on this next slide, and this is also different than that we did with Professor Mielke at Mayo Clinic, where we showed that if you compare the Lilly assays and the Gothenburg assays, that basically for diagnosis, then you don't see much of a difference. They tank together, and these are the ones that if you're just looking at age and sex, but then if you're looking at just the biomarkers, where these are the colored ones, then the difference, you don't see much difference between them, no significant difference between them. But then if you start to look at it for like predicting A beta positivity, then you can see like some tiny difference that appear. So this is, to me, is a good thing to show that we've come to a stage that although we have been quite would say like very spoiled with choices, and it comes back to like, when there was the COVID vaccine situation, I always link it to it, that when COVID struck and we were all concerned and there was no vaccine, we're all voicing out our complaints. But then when the vaccine started to roll in, and then there were more than one, then we started nitpicking, which one do we want, which is better than that? This has 90% and the other one 85% accuracy, and that's the same thing here. But to show that for diagnosis, it seems you could use any of them and you get the same results. But then if you want to look at something else, then you might have to choose a different one. So in terms of clinical performance or clinical use, what do we think? How should we use them? We proposed this algorithm and of course it's always open to questions and to interpretation, that we think that one thing, that the biomarkers should not be used by themselves, that we're not going to create a whole new paradigm that, oh, that biomarker just by themselves. No, they should be integrated to what exists already. It's going to come in to help the clinician make a decision, maybe faster, maybe more accurate, but not as a standalone. That you could integrate that, where if someone comes in with a cognitive complaints, you go through the normal algorithm, what is done, and that instead of going through the regular approaches here, then you can add in a blood analysis. We're proposing that we could start, for example, phosphotel because of its specificity, and then you could come into the results based on, say, okay, this is kind of like a traffic light approach. If the results like low, then there's no problem, or there's little concern. So then you can try and link it, try to measure NFL, which you look at degenerative causes, neurodegenerative causes. If it's high, then there could be concerns of non-AD neurodegenerative concerns. If there's nothing also there with NFL, that both are negative, then you could also look at lowering down the concern and looking at monitoring the person at the primary care, for example. But then also, with all these things that we're talking about, there's one thing I say, it seems quite easy, but then it's going to be difficult to do. That, for example, with this traffic light approach, how do we develop it? We have to develop cutoffs. Even if you want to go with two instead of three, you have to develop cutoffs. And at present, in which data sets are we going to develop those cutoffs? In which populations are we going to do that? In which age group are we going to do that? What would be the demographics that people will do, that we test them in? In which assays are we going to do? Because now there are different assays entering and potentially going to enter the clinic. And these are questions that we have to answer. And this is what I want to try and focus the rest of my talk on, to say what is left to do and how potentially should we do it, just to raise questions that we can all go away thinking of how to answer and how to try to integrate those in our research planning. So, for example, the first thing, and this is a question that we genuinely ask ourselves here, that when there's phosphotel increase, what does it mean? Because we say, okay, phosphotel increases, it correlates really well with A beta pathology. For example, we show like 231, 181, 217, they're all increased. But then if you like according to A beta pathology, the A beta PET changing. But the same biomarkers also correlate really well with tau PET. So primarily, what is phosphotel? Is it in blood? Is it a CS? Is it an amyloid marker, or is it a tau marker, or is it both? And if it's both, that raises concerns. For example, reason like in clinical trials, are you going to use if you're using an amyloid trial, and if it also signals something with tau, could there be a concern there, or vice versa also? And these are things that we have to try to answer. And one way to do this is to go to animal models. And one of the papers that I quite like that came out last Christmas was to show in a mouse model of amyloidosis, overexpressing APP, which is the protein that produces amyloid beta. You could see that both phosphotel 181 and 217 were increasing quite well in those animals. At the same time, in a mouse model, and this is a model that doesn't produce tangles. At the same time, in a mouse model for the Danish variant of the amyloid producing protein, you could also see the same thing increase. And both models did not produce tangles. So it means you could have phosphotel increasing in the absence of tangles. And I think one thing, and I'll come back and talk about that the blood phosphotel that we're using currently now, it kind of like biochemically not exactly the same as what we have in CSF. And that's one reason why it starts to appear quite early, like in response to amyloid, for example. So this is one thing that was suggested to say, okay, that in a recent paper to say, okay, it seems like A beta deposition is the first thing that triggers phosphotel increases, and that would also lead to increase in hyperphosphorylation and being released into biofluids, and then you could have some going backwards, and that could lead to cognitive decline. But then because that's the question that I'm trying to answer, and I think it will be good that we all try to answer that. When you have phosphotel increase, because if we're going to the clinic with these markers, we should first be able to understand when someone has phosphotel increase, what does it mean? We don't know. And for example, so we tried like in a primary care settings, for example, more recently, and what we found is that in a primary care setting where you only have people, been preliminarily diagnosed based on MCI or AD, or if we have like young individuals or older adults who supposedly don't have cognitive impairment. All that we can tell is that the marker levels, this was 231 and also 231 and then 101, the marker levels are high. But then being high, they are high in both who are AD, who are preliminary AD or MCI or elderly. But then if you don't have A beta levels, then which one, for example, if you based on these two plots, which ones do we decide to take them to the next steps? First of all, what does it mean? Is it like we could say, sure, with certainty that they have AD, or could we say that they have amyloidosis and that they should be monitored for what happens later? And how is that going to agree with the clinical diagnosis or with the clinician's decision? And what cutoffs are we going to use, as I talked about initially? So this is a proposed way that we first have to figure out what an increased phosphotel is. If we say phosphotel is abnormal or is increased or is changed, what does it mean? We have to figure out what that is and how it's going to influence clinical decision. And if we're going to say that's like a marker for amyloidosis, we have to be able to figure that out, and how we confirm with CSF or PET markers as well. So just a quick overview of that. That, for example, phosphotel or tau itself has physiological functions beyond the pathophysiology that we know. So we have to be able to figure out the changes across the age course. Now, for example, it tends to have some neurodevelopmental changes when it comes to like umbilical cord changes. It drops and then it tends to pick up later as well. So do we need age adjustments? We don't know, because we haven't really monitored the biomarker across different age groups. And that also, we have to figure out what biological factors could affect the levels. Do we have sex effects? Do we have any sort of epigenetics, any comorbidities, any conditions that could affect it as well? And in terms of lifestyle, if someone is using a specific kind of medication, do we have to monitor them for that? And then factors that affect tau metabolism, liver disease, kidney disease, because we know these organs are places where tau is also expressed quite a lot. There's also one thing I have to look at. So just a quick example that we looked at race and ethnicity in with phosphotel more recently. There've been a few papers in the past talking about CSF and all that, but I personally was a bit cautious in terms of the numbers. So what we tried to do, we partnered with Dr. Schindler at WashU, where we looked at individuals, 76 pairs from the WashU ADRC cohort, the night ADRC cohort, and they were paired for these. So basically for each African-American person was paired to a non-Hispanic white person of the same age, the same sex, the same cognition, the same ApoE genotype. So very stringent ways. And then they sent the samples over, and I ran these ones myself in a very blinded way. And what we did find was that when you feel like first replicating what had been shown in CSF, that for the CSF A beta ratio, you tend to see like the levels did not match exactly. And then also in plasma phosphotel, plasma A beta ratio using the C2N method, it showed something similar. And then with amyloid PET also, it shows something similar, because it's always going to be the other way around, but it shows something similar. Basically, so that it's suggesting to me that in this set, and I'm quite trying to be specific, in this set of individuals, the African-American set had lower levels of amyloidosis compared to the non-Hispanic white set. This is quite it raises a lot of questions and worth to study. And based on this, when you use plasma phosphotel, whether we tried 101 and 231, although statistically there were no difference, but then the ability to predict increased or to predict abnormal CSF A beta ratio or abnormal amyloid PET in regression models, that it seemed to provide a bias against the individuals who were in the African American group, showing there's a lot more for us to all study and to understand that what these markers mean, and if they work the same way for everyone before we start to roll them out in on large scale as well. One thing that we have to look at also is robustness, like how robust is the marker, because what we've done so far has been nice, but then they've been done in I would say well controlled settings. Like if we're doing things in the lab, we can you have the same protocol that you can follow. But then if you go into the clinic, which means that it's going to be multiple centers running these assays on different instruments at different times of the day, and might be that the protocols would vary, then we need markers that are very robust. We ran this analysis recently in Nature Reviews that we just returned the proofs for, so that should be coming out soon. And basically what we saw was that if you have CSF, A beta 40 ratio, and then we plot according to the density of those who are positive and those who are negative, then basically you get to see that CSF A beta 42 is like, the best thing. It can separate quite nicely. But then the plasma was not the best. Like the fold change was really small, less than 10%. So it means that anything that can affect this stability, this robustness, is going to mess things up quite well. So that kind of questions, in terms of the widespread use in terms of for plasma beta, and we did not see the same thing for phosphotel. And then in another study, just showing the same thing, that we modeled that if you look at all these biomarkers in the same set of individuals, and it's okay, these changes. So like when you run a study, if you have, let's say, 1,000 individuals, 1,000 samples, you cannot run them at a go. You have to run them at different times. And that the variability between those runs is something that can affect your results. That variability is expected. A small amount of variability is expected, but of course, the larger, the worse. So we modeled that, for example, if you have variability changing between 0 and 20%, that the marker that was affected the most was the mass spec A beta. In agreement with the small fold change that I just talked about. That if you're picking at if you're able to have the variation to be very small, so no variability between runs, then it's a very excellent marker. But then if there's a little shift, then it tends to affect its performance really well. And these shifts could be day-to-day changes, or your reagent batches changes, or something that you bought from the commercial source, just changing a little bit, that could lead to these aspects. And of course, pre-analytical handling is also another important aspect, that the way you design your study, the way you collect your samples. For example, we know A beta binds to tubes and things like that. So you have to really think of this from the very early stages when planning a study. If you're going to send results to someone like in a different lab, you have to look at what conditions you're going to use. And even before then, you have to look at what conditions you're going to use to store your samples. Are you going to store in -80? Is that -80 also very alarmed, for example, so that if there are any changes in the temperature, that you can pick it up quickly and take good care of your samples. But then one thing also, and one thing I like to do a lot, is to think about the unconventional things. That we're very good looking at. Someone comes to the hospital, you take blood and you process and you go through the normal approaches, but this is not realistic everywhere. At least the pandemic showed us that this is not realistic even here. So we thought of different ways that, when it comes to remote sites where shipping is not a good thing, and then where you can't even have reliable electricity to store your samples, we have to start thinking of how we can bring blood biomarkers to these places, and to make sure that everyone benefits from that. So we started with neurofilament light, and we tried these 3 approaches. That there's this Noviplex card that basically collects whole blood, and then you put on that spot here, and then the separation occurs on the card. Or you could try a normal dry blood spot. Basically, you just take a prick, and then you drop the blood, and then you're on normal filter paper, and you allow it to dry, and later you put it in a diluent, and then you let it dissolve, and you can spin it. Or mimicking like a field work, where you go to somewhere remote, you take your samples, your whole blood, you just leave them at room temperature, you bring them to your center in process, so that we allow 48 hours, so that we allow 48 hours before processing the blood samples, and compared against the standard of truth. And that standard of truth is that if you process the blood, they just immediately as you collected it. And that is what we found, that compared to the standard of truth in 2 very small cohorts, this one and that set, in both sets, in this condition and that condition and that condition, and also in a first set of cohort and also a confirmatory, we have very good association agreement between these and the standard of truth, although the absolute concentrations, of course, are going to vary. But at least it shows one thing, that maybe we can start to think of using these markers in settings that we cannot have everything nicely done. Close to ending, so if we're looking at amyloidosis and how these can also, if we say amyloidosis, if we say phosphotel increases is a measure of amyloidosis, then how do we define amyloidosis? We know that if you use CSF A beta 42/40 ratio, it changes slightly earlier before A beta PET. So the one that you use to define your amyloidosis is going to have an impact. If you use CSF, also like CSF is going to be a little more stringent, and amyloid PET is going to be a little more liberal, but the one that you use also has an impact. And if you use plasma as well, plasma A beta that we don't have so much about yet. And then that's this one also, because one of the changes, the effect is that how it can affect how you predict your amyloidosis, like how well your plasma A beta phosphotel will predict. So this is an example of what we did in a recent paper. This is like in CSF, if you're using CSF A beta 42/40 ratio as your marker for amyloidosis, your standard, then the ability of phosphotel to predict abnormal amyloid pathology is less compared to if you use A beta PET. So this is going to have an impact as well. And how is all this going to affect clinical decision making? I created this just to say, like, there's a puzzle that we have to think of solving. That if we have like plasma phosphotel levels here, and there's a cognitive impairment, assuming everything else stands the same, like all the clinical assessments stand the same. If someone has a preclinical disease, they're going to have increased phosphotel, but then their cognition is going to be intact, so no impairment. So I guess it's going to be easy, or comparatively, to pick that one out. And if someone also has a late dementia, then, which is not of the AD type, then you might also pick it quite well. Like, phosphotel will be low, cognitive impairment will be there, but it might point to non-AD potentially. But when we have people who are normal age and those who have clinical phase of the disease, that's where one of the phases that we have to try to figure it out as well. And this, of course, has to integrate with all the other complexities that have to happen in the clinic. And lastly, we have to confirm with CSF or PET. The few papers that have tried to do that, this was in the JAMA Palm quiz paper, although it doesn't there's a lot of the data sets are quite packed here, but at least it shows that you have some individuals who show positivity for plasma phosphotel. This was 217, but then their CSF phosphotel was still low. So it just will show that it's not all the time that we're going to have good interchangeability. And we saw the same thing also with phosphotel 101. You have like a quadrant, basically. Some individuals have good agreement up here and down there, and others do not. And these are ones that we have to try and figure out, and that also showing the same thing. And there's one paper showing that question of but why are they not interchangeable, and what affects this interchangeability? Because there are papers showing that we can really CSF and plasma are basically the same. And I think the reason is that when it comes to this famous figure in the JAMA paper showing that when you predicting based on AD dementia versus non-AD dementia, then these markers will perform almost the same. But then when it comes to the other phases of the disease, where you have subjective cognitive decline or mild cognitive impairment, that's where you might start to have some disagreements. We can have those who are either A beta positive or negative, and those in the gray zone of positivity, and some other heterogeneous presentations of diseases are also going to affect them as well. And just one thing that also affects this interchangeability is because of CSF versus plasma, is because currently the assays that are in the clinic approved for in vitro diagnostic use for CSF, for example, they tend to target the mid part of the region of the tau protein. And it's something that I started to figure out from the start that really pointed me to this direction for plasma phosphotel development. It's very reassuring, and this is something that's in a review coming out. It's very assuring that basically all the phosphotel assays from all the different sources that were developed independently are targeting that same region, because what we know is that this is where the regions in blood, you get to have more of the N-terminal parts being retained, and that you don't have a lot of the C-terminals in that you tend to see in CSF as well. So last part, when you compare with tau PET also, phosphotel picks up earlier than tau PET. that probably it's understandable because the soluble part will show earlier than the PET part, which is looking at aggregates, and which they have to clamp up before you can see changes. So then this also has implications if we're going to use phosphotel as a surrogate for tau PET. There are things that we also have to start to think about. So in conclusion, we've covered quite a large breadth. That the blood biomarkers are super high promising, and they're so exciting for us as a field, but also even more importantly for patients and their families. But then, as we go into the stage of rolling out in the clinic, maybe there are a lot of questions that we have to understand and we have to try to address whilst we go to the clinic and try to use them in all kinds of situations. And thank you very much for indulging me and all the massive group of people who have been our backbone. We thank all of them, and thank you for your time.
Thank you, Dr. Karikari. That was a fantastic talk. We got super excited about blood biomarkers, and then we got super cautious. So thank you for both of those perspectives. We're now open for questions. So if you have a question for Dr. Karikari, please feel free to make your way to the microphone in the center. Tell us your name and ask a question. We're also taking questions from the individuals who are joining us online. Do we have any questions or comments?
We do have a question from one of our online attendees. They start with a compliment. This is very exciting work. Thank you. Can you comment on if any of your phosphotel biomarkers can significantly differentiate MCI from cognitively normal cohorts?
Yes, I think they do, and but it also depends on if we're looking at MCI and then cognitively normal group as just by the clinical diagnosis, or if we're also going to separate them out based on the A beta positivity as well. But then what we know is that most of the time the markers in MCI stage, those who are A beta positive have increased levels compared to those who are preclinical disease, like cognitively normal, but then also with A beta positivity, and that's also higher than those who are cognitively normal but no A beta. So I think the performance of these markers will be very much dependent on whether you're separating them by A beta or not. Similar to what I showed, that in a snapshot of primary care, that you get to see that you can separate out the MCI from the AD because the level, if you don't have any way of separating out by A beta as well.
Thank you.
Thank you so much for this mind-blowing overview of questions that you've left us with. No, you also had a lot of answers. Thank you so much, Dr. Karikari. I would like to add to, I found it very astonishing the differences that you showed in the biomarker levels for the with the racial disparities, basically. Do you think Alzheimer's disease is going to be a disease where we are actually going to develop cutoffs that are going to be very specific? And can you maybe comment on so you matched for genotypes, so APOE, cognition, sex, and another factor. Can you comment on what you think is what the underlying differences are that lead to these differences in biomarker levels that you saw?
Yes, yeah. This is always an interesting, but also very, I would say, concerning aspect. But then first thing, in terms of race-specific cutoffs, it's something we definitely should stay away from, in that there hasn't been good precedence of that in other areas of medicine. Like, in neuropsychology.
I was wondering about that too. I mean, there's no specific markers for diabetes or something like that. We have one cutoff for the biology of the disease.
Exactly. So one thing that we found in this data set is that it looks like you're one of the reviewers for the paper. Yes, you're asking now the questions the reviewers did ask. That we looked at, for example, when it comes to diabetes, and then there was a higher diabetes proportion in the African American set, and also a higher hypertension also. But then no one has been able so far to link these aspects to biomarkers specifically. And this is one thing that I'm also interested to look at, that if we could start to look at a wide variety of populations, people who identify, and I try to use the term identify because of how fluid these social constructs are, and then trying to see that, is it that we are finding something, because we don't see any biological sense there in terms of race, but could it be something social, because there are lifestyle differences that could link to these aspects that we do not understand as well.
Thank you. That was really inspiring.
All right. Thank you so much, Dr. Karikari, for your presentation. We will let you get on with your evening. Thank you for joining us so late, but it was quite the honor. Thank you very much.